This next section talks about leukocytes and thrombocytes. So leukocytes are white blood cells and thrombocytes are platelets. And this shows kind of the differentiation of the different types of blood cells. And this entire process happens inside of the bone marrow. So you start with a hemocytoblast, and then that turns into a proerythroblast, a basophilic erythroblast, a polychromic, a reticulocyte, and eventually the erythrocyte, which leaves the bone marrow. From a myeloblast, a promyelogranulocyte turns into this basophilic myelocyte, an eosinophilic myelocyte, or a neutrophilic myelocyte, which eventually turn into the basophils, eosinophils, and neutrophils. A lymphoblast becomes a lymphocyte, a monoblast becomes a monocyte, and then megakaryoblasts become megakaryocytes, which eventually fractionate and become platelets. So there's two major categories of white blood cells. Excuse me. The granulocytes, which contain granules, and the agranulocytes, which do not contain granules. The granulocytes include neutrophils, eosinophils, and the basophils or mast cells. So these all have distinct shapes to them and distinct features. You'll notice with neutrophils, usually you'll see multiple lobes to the nucleus. Eosinophils usually have these dark red granules, and basophils usually have dark purple granules. A neutrophil is a major phagocytic cell. So these guys are going to eat debris and foreign microorganisms that come into the body. If you have a bacterial infection that ends up producing pus, it's the neutrophils that are making up a majority of the pus. And you can see this if you look at a pus smear under a microscope. Most of the cells inside of a smear of pus are going to be these neutrophils. An eosinophil plays a major role against parasitic infections and during times of inflammation. Basophils and mast cells also mediate inflammation, but they play a role in allergies as well. Agranulocytes include the lymphocyte and the monocyte or macrophage. And before I forget, the basophil and mast cell, basophils are what we call them when they're in circulation. Mast cell is when they eventually settle in the tissues, kind of like with the monocyte macrophage. Monocytes are when they're in circulation, macrophage is when they're settle in the tissues, but they're basically the same cell, just in a different location. The lymphocytes function is, it makes up your, so these are your T cells and B cells. This makes up your adaptive immunity, your third line of defense against pathogens. These cells are smaller and their nucleus takes up a majority of the cell. Monocytes and macrophages are also major phagocytic cells. While the neutrophils primarily eat bacteria, monocytes and macrophages eat debris. So with a monocyte, it's a huge cell and it has a lobed nucleus. Macrophages are flattened cells and you'll see them in the tissues. Macrophages are called all sorts of different things depending on the tissue that you're finding them in. A differential white blood cell count uses a flow cytometer machine in order to give you a count of how many of each type of white blood cell is found in a patient's blood sample. This blood that you run through a flow cytometer cannot be clotted. So it has to be unclotted blood. You would spin it down or centrifuge it to separate out the different layers of the cells, and then you would run it in this machine, and the machine would tell you relative cell counts. A differential white blood cell count is usually, like, the machine is usually followed up with a person actually counting the different types of white blood cells that they see in a blood sample. Those differential white blood cell counts can help diagnose these types of homeostatic imbalances. So leukemia is a cancer of the blood cells. Usually in leukemia, you have an excess number of white blood cells, and they're usually immature white blood cells. So there's some type of blast cell that you're going to find in excess in peripheral blood. SIDS, severe combined immunodeficiencies, is when you're completely lacking T cells or B cells. So that leads to a decrease in production of those cells. Neutrophilia is too many neutrophils. That's usually in the case of bacterial infections. Neutropenia is not enough neutrophils. Eosinophilia is too many eosinophils. Eosinophilia can be a sign of a parasitic infection. Eosinopenia is not enough eosinophils. Basophilia is an excessive number of basophils that can be present during times of allergic reactions. Basopenia is not enough basophils or too few basophils. All of the philias can also indicate types of leukemia. So now switching gears to platelets. Platelets are called thrombocytes. They're small pale bodies that appear as irregular spindles or oval disc. Megakaryocytes. So megakaryocytes are these large cells. And a megakaryocyte, so that's the nucleus, will fractionate off. So parts of its cytoplasm will kind of chunk off. And that's what a platelet is. It's basically a chunk of a megakaryocyte. Platelets play a role in agglutination, or clumping, adhesiveness, or sticking to tissues, and aggregation, or calling others to the area. So they can actually release chemicals that call more platelets to the area. So if you cut yourself, the platelet plug will form. An injured vessel is a portal of entry for bacteria, and there's also damage to the cells of the vessel itself. That stimulates the adherence of platelets to the area of damage. When platelets stick, they're going to release chemicals that call more platelets to the area. And when those stick, it's kind of a positive feedback mechanism where more and more and more platelets are going to build and build and build. Eventually, sticky platelets form a physical plug and secrete chemicals involved in the coagulation process. Blood clotting. So there are clotting factors within the blood that will promote the formation of blood clots. When blood clots form, they can either be localized or they can be traveling through the bloodstream. A thrombus is a localized blood clot. So that's when a blood clot is blocking a particular blood vessel in one region. If that blood clot dislodges and ends up circulating around in the body, then we call it an embolus. So an embolus is a blood clot that is moving through the bloodstream. That embolus can then be clogged somewhere in the body. And if it gets clogged there, it causes excessive tissue damage and suffocation to the tissue distal to the clog. One of the most common types of embolisms is a pulmonary embolism. If you think about the flow of blood through the body, so you've got a person, and then you have their heart in the center of their chest. Yes, I know I'm a terrible artist, but let's say that this person had a blood clot in their leg, which is one of the most common places for a blood clot to occur. The blood clot can form inside of a blood vessel, so usually in a vein where the pressure is a lot less. And if this blood clot dislodges, it's going to go through the cardiovascular system up the vein side of things. So from the vein, it's going to go into the inferior vena cava, into the right atrium, through that tricuspid valve into the right ventricle, and then off to the lungs in order to get oxygen. When it works its way into the lungs, the blood vessels are going to get smaller and smaller and smaller until they lead to capillaries. Well, that is where you can have a blockage happen. So that blood clot from the leg can end up blocking a major blood flow to the lungs. That decreases the ability of the lung to properly respirate. So oxygen and carbon dioxide cannot be exchanged if the blood flow to the area is blocked, and that can lead to death. So pulmonary embolism, again, these blood clots, and they can exist anywhere in the body. But when they go back to the heart, the next place that it's going to get really small is going to be in the lungs. If they are suspected to have a pulmonary embolism or any type of stroke or anything like that, or if they've just had a heart attack, and we need to loosen up blood flow a little bit, we do have anticoagulants. So warfarin blocks vitamin K, heparin blocks thrombin, which is a clotting protein. Aspirin prevents the production of a molecule that causes platelets to aggregate. It's important that you know the names of the major anticoagulants, but not necessarily their specific function. So some homeostatic imbalances associated with thrombocytes. Thrombocytopenia. So penia, remember, is not enough, so that means that you don't have enough thrombocytes. So it's a clotting disorder due to a lack of platelets. What you'll see on the patient is petechiae, and you can see that on the surface here, like little bruises or blood clots under the skin. Gastrointestinal bleeding and melena, or blood in the stool. Melena is a black stool due to bleeding that happens early on in the digestive tract. And hematuria, or blood in the urine, and all sorts of other stuff. Then there's hemophilia, and hemophilia is an X-linked inherited bleeding disorder which leads to the failure to produce clotting proteins.